Xtalks is the industry leader in connecting life science, food, healthcare, and medical device professionals. The Xtalks Life Science podcast features insightful conversations with industry experts about all things pharma, biotech, and more.
Hello and welcome to the Xtalks Life Science podcast. I'm Vera, editor-in-chief at Xtalks.com, and this week I'm joined by a very special guest on the show. We have with us today Dr. Carsten Rudolph, CEO and co-founder of Ethris, which is a biotech company focused on developing innovative mRNA therapeutics for the treatment of asthma and rare lung diseases. His expertise is delivering mRNA specifically to the lungs. He is the inventor of 15 patents and applications and has authored more than 120 scientific publications. Dr. Rudolph, thank you so much for joining us today.
Hello, Vera.
So to start us off, as the co-founder of Ethris, can you share the initial vision for the company and how it has evolved since its founding?
Yes, of course I will do. Basically, it goes back to my academic times, and I was doing research at the university in Munich and in the pediatrics department. And I was always looking, into finding potential treatments and potential medications for patients with the focus on pulmonary diseases. And in these days, we looked into potentially also using messenger RNA, and we found that messenger RNA indeed offers many advantages over other approaches that were used in those days. But what we thought is, well, if we don't start to think about commercializing it and developing this will basically end up in a draw. And so that's why I thought with my partner and co-founder Christian Planck, we just have to found a company, and that's where we started the company and where we set up the company. So that's the reason behind it, why we started Ethris.
And what year was the company founded?
That was in 2009, basically, and we became operational. It took us some time first to raise some money and to get everything sorted, and became fully operational towards the end of 2010 and 2011. Then we became fully operational, so about 10 years ago. And so we are very happy now that we have our first program in the clinics. It was a long way, but this is sometimes what you need if you do research.
Yeah. And what initially sparked your interest in targeting the lungs with mRNA technology?
That's indeed a good question. And also this goes back to my academic background. Like I mentioned, I was heading a research team at the Department of Pediatrics here in Munich at the university. And as you know, cystic fibrosis is a quite common disease in especially hospital for kids. And so that's why always my research centered around cystic fibrosis. And this is a lung disease. So that's why I was always in lung diseases. And that's so historically, basically the reason why we started the company, why we looked into developing mRNA Therapeutics for the treatment of lung diseases.
Great. And could you talk a little bit more about the company's mRNA technology platform, how it works, and what may be the potential impacts in the therapeutic space of lung diseases?
Yeah, so this is important, and because and I think you're familiar also with their pulmonary drugs. Pulmonary drugs always need to be inhaled. That means delivered via a device. These asthma sprays basically that patients use. So what we did at Ethris is really to tailor messenger RNA delivery around this challenge. And I say it's a challenge because it's not an easy drug format. So what we have developed at Ethris is our critical elements. First of all, it's very important to consider the stability. That means that these are tiny nanoparticles that are used for packaging the messenger RNA, that they are not destroyed during the step after inhalation. Yeah, so we did a lot of work there, and we have a solution for this, so that we have developed a stabilizing excipient that fully prevents any aggregation that occurs or could occur during nebulization, on the way to the lung. And you may recall that this is also really an issue from the commercialized vaccines, where basically even on the label it is written, please don't shake, because these particles aggregate. And of course, during nebulization, the stress that acts on the particles is much, much stronger. So that took us some time, but we have developed now a technology that fully prevents any instability during this step. Yeah. The next element, of course, that is very important is that the formulation, the nanoparticle itself, so that it is capable of delivering messenger RNA into the epithelium of our lung tissue with a very high efficiency. So we did a lot of work there and developed a very strong technology. Yeah, and then, of course, what is important, we always, in the end, have the desire to provide to the patient a very easy to use and very convenient drug products. And if we look forward here, this is these easy spray dry devices that are used by these patients. And one precondition there is that you can store it at room temperature. Yeah, so we did a lot of work to stabilize our LNP formulations at room temperature. And so we developed a lyophilization process. We have now stability for more than 15 months, which is, we believe, really quite differentiated in this space. Yeah, and more importantly, we have also now achieved spray drying of our formulation to create really powders that can be inhaled. So that's a very important step that we have achieved here. And on top, we have also developed our technology to make it better controllable. And what I mean here is that their LNPs, these nanoparticles, after delivery to the lung are retained only in the lung tissue, so that they are not absorbed basically into the blood circulation, but they are really kept at the site where we need the potency, the activity of the drug product. Yeah? And with this, we can, of course, also minimize potential side effects. So we have a full control of our LNPs in the lung tissue. Yeah. So, and all together, because of the stability and control, we call them SNAP. Yeah, that means stabilized nanoparticles. Yeah? But this is more on the formulation side, We also did a lot of work on the payload itself, which is the messenger RNA. And so we have developed a technology to modify the messenger RNA so that it's not recognized by the sensors of our innate immune system. Yeah? So that we don't have an inflammatory response. Yeah? And you know this maybe also from the commercialized vaccine, there's also a modification technology used. We do this a little bit different, yeah, but achieve exactly the same properties. Yeah? But we also have there, I think, an advantage, because with our technology, we can avoid potential frame shifts. That means the production of undesired proteins, which has been observed for the technology that is used in the commercialized vaccines. Yeah? And then if we look further into our technology that we've developed, is also on the manufacturing side. So we have developed technology for large-scale manufacturing that is built totally or purely on tangential flow filtration, which is a very easily scalable manufacturing process used as a standard in industry, and does not need HPLC, which is a very time-consuming process and otherwise used as a standard in the field. So that was also very important technology that we've developed at Ethris. So it's really about Storage, easy storage at room temperature, yeah, which is important for our drugs product. It's about stability, yeah, which is very important, and it's about efficacy, yeah, which is a key criterion, and it's about local control. That means really retention at the site where you need the drug, and no distribution beyond.
Great. I was just wondering, could you comment about how that stability and efficacy, how does that compare to other mRNA therapeutics, like in development or perhaps already on the market? Do you know what I mean? Because you mentioned stability at room temperature for 15 months, I think it was.
Exactly, yeah. So I mean, the data that I know is, and if you look at the commercial vaccines, yeah, at room temperature, they have to be used after a few hours to a day, yeah. And so that's why I believe that we really have made a step forward here with our technology to make it more user friendly.
Yeah, for sure. And let's talk a little bit more about the indications that Ethris is focusing on. So the company's current pipeline includes candidates for asthma, but also primary ciliary dyskinesia and pulmonary alveolar proteinosis, which is also called PAP. Could you elaborate on what are the unmet medical needs for each condition, and how and why Ethris's R&D efforts are aiming to address them?
Yeah, that's a very, very important question, Vera. And let's start with asthma. And asthma, and I think that's not known to many people because we have so much medication on the market. But it is the current medication is not sufficient to really fully control the disease. And what do I mean by this? That means we still have exacerbations occurring. Exacerbations, are these typical asthma attacks, that patients suffer from. That means you all of a sudden have shortness in breath. Yeah, you grasp for breathe. You have tightness in the chest. You have coughing. You have wheezing. Yeah, you have awakening in the at night because of asthma. Yeah, and what is important to know is that each of these asthma attacks, leads to a decline in your lung function, which is not entirely reversible. Yeah, that means the more asthma attacks you have during your life, the lower the lung function gets, and this is really a problem that still needs to be solved. Yeah? And in this context, it is important to see that we have really many patients. Just in the US, we have about 4.5 million patients with uncontrolled medium to severe asthma. Yeah? And this leads to about 1 million emergency department visits and even 100,000 hospitalizations. Yeah? So there's really a great need from the healthcare resource utilization, but also for the patient, there is a burden. Yeah, you can imagine that a dramatic event of an asthmatic attack has also a psychological component, behind it. So there's really a high unmet need. And what we do with our candidate ETH47, which has now completed phase 1 trials, we address these asthma exacerbations, because you have to know in about 80%, these exacerbations, attacks, are driven by infections with very harmless viruses, like a rhinovirus, where we both just get a runny nose, and a cold. This can trigger an exacerbation in the asthma patients, and we don't have anything there right now. Yeah, and with ETH47, what we do here is we deliver a messenger RNA that encodes for a type 3 interferon. Yeah? And these interferons play a central role in maintaining the resistance of our epithelium against the virus infection, so that the virus does not spread in the nasal cavity or even spread to the lung tissue. And this molecule, interferon lambda, is even dysregulated in asthma patients, and also with a delayed response. So what we do here is that we want to provide to the patients a once weekly nasal spray, to produce interferon lambda in the nasal cavity and induce a broad antiviral status in the epithelium, so that any potential invading virus is not capable of replicating and further spreading in the nasal cavity or even further down to the lung tissue. So to fully control and prevent virus-driven exacerbation. So it's a very unique, new, and potentially also disruptive mode of action. And this is it does not only hold also true for asthma patients, but this is also observed to a lower extent also in COPD patients. Yeah, so there's a broad utility of the drug product candidate, we believe, in the future, and we can even also think about, and you have this in the US, what I saw in the press with this new bird flu H5N1. Yeah, so let's think also potentially into the direction of pandemic preparedness. Yeah, where we know that the potential virus X will emerge in the future and could lead to a pandemic again. So if we had something available with a very broad, virus independent mode of action, we may also have something that we can provide in the future, in particular to patients, to people that are critical in our infrastructure, like medical personnel, or to a very vulnerable patient population, to gap the time, to bridge the gap until we have then tailored vaccines available. Yeah, what we learned from the last pandemic takes something like a year, but more than a year. So it really has great utility, this drug candidate. And then also regarding, you mentioned primary ciliary dyskinesia. Yeah? So these patients, they suffer from mutations in genes that are responsible for the beating of our tiny small hairs that cover our airway epithelium, and that is responsible for transporting the mucus out of the lung. Yeah, so you can imagine if they are non-functional anymore, you have a mucus congestion in the lung. And this is exactly the symptoms that these patients suffer from, a little bit like CF patients. That means that they have a chronic wet cough. Yeah, they have a chronic rhinosinusitis, also frequently otitis media. So this in the end leads to chronic bacterial infections and a decline and progression of the lung function over time. And we don't have anything for these patients available right now. So, and here the concept is to deliver a messenger RNA that is encoding for the missing protein of the patient to restore the cilia beating again, so that we have an active transport of the mucus again in these patients. Yeah? So that's the concept behind it. And similar concept is also for our drug candidate ETH45 for the treatment, and you mentioned it, pulmonary alveolar proteinosis, which is also a very rare disease, and where patients suffer from an autoimmune disease. So these patients develop autoantibodies against their endogenous cytokine called GM-CSF, and which is responsible for macrophage activation, which is mediating the homeostasis of our called surfactant layer in the lung. Yeah? And if this is not functional, then we have a buildup of the surfactant layer deep in our lung, yeah? Which prevents a proper gas exchange in our lungs. And in the end, these patients suffocate, yeah, and we don't have any treatments available for these patients right now.
Well, thank you very much for sharing for each of those three conditions, and I really liked how you explained kind of like the biochemical reasons of why mRNA can help for each of these three lung diseases. So that was very insightful. I wanted to ask you, as Ethris is now, advancing these novel mRNA therapies into clinical trials, could you share what are some of the key regulatory and clinical challenges you anticipate there may be, and how is the company preparing to address them?
Yeah, that's always a good question. Yeah, so I mean, as with any new drug modality, we of course face challenges of a developing regulatory framework. Yeah, so that's of course clear. I think it's better for us now because with the approved mRNA vaccines, yeah, so the field got a lot of tailwind. Yeah, and but more, and we can learn and take the learnings from that field also in our development. Yeah, so that's very important what our colleagues did in the space here. But for us, if we look into asthma, maybe one limitation is that, and this is the next step, of course, in our phase 2a study, we want to test the efficacy of our candidate ETH47 in a very controlled setting, where we challenge, that means infect, asthmatic patients with a rhinovirus to see in a controlled setting that our drug is exerting the effect what we want and what we expect it to do. Yeah, and these challenge studies, there are only limited geographic areas where you can do this. So it's a very specialized CROs that you need there. So that's a little bit where we deal with a more limited choice of specialized CROs. Yeah, and in PCD, it's like for any rare genetic disease, it is about awareness, yeah, and working with patient organizations together to find the patients basically for your clinical trials. Yeah? So, but I think also there we have learning and precedents from other genetic diseases like cystic fibrosis or from other metabolic or genetic diseases. But these are some of the challenges that we have in our development path that we see.
Well, thanks for sharing. And you're right. You have mentioned that post-COVID, there are now many companies developing mRNA therapies. How is Ethris differentiating itself in this competitive mRNA landscape right now, particularly in the respiratory field?
Yeah, this is technology, I would say, and that's what I mentioned already before. It is really our really differentiated pieces of technology. So that's our stabilized nanoparticles, our SNAP formulations, with a very unique cationic lipidoid based technology for very efficient delivery of messenger RNA to the cells of the respiratory system. Yeah, there are unprecedented, I think, stability of our formulations, which is a key challenge. I mentioned this already before for delivery, in particular to the respiratory tract, and also storage. That we have really achieved already very good long-term storage at room temperature. Yeah? It's a very key element to have a successful drug in the end on the market that has a high rate of acceptance by our patients then in the future. And it's safety. That means control of your formulation so that you can fully retain the activity of your mRNA drug at the site of delivery and avoid any spillover into the blood circulation or any secondary organs. So those I would say are really key elements with our differentiated modification technology, as I mentioned before, to minimize any potential interaction with the immune system and avoid also potentially frame shifts that has been observed for other modification or the commercial modification technology that is used in the field.
Great. Thanks for sharing those specific areas where Ethris is kind of making a difference. And as mRNA therapies are gaining more and more traction globally, in your opinion, where do you see the biggest opportunities for Ethris to make an impact in healthcare over the next 5 to 10 years?
Yeah, so I think those are our areas where we are active in, and like I mentioned, for asthma, we have a high unmet need. We have an unsolved problem to fully prevent exacerbations and to really achieve a clinical remission in these patients. And if we just think about the pure size of asthma patients we have globally, so it's something like 260 million people worldwide suffering from asthma. There is a big unmet need that we want to address in the future. And I mentioned before, this is not only restricted to asthma, but also a large proportion of COPD patients suffer from these virus-induced exacerbations. And I mean, we also have globally about the same size, so something like 300 million patients suffering from COPD worldwide, and it's increasing the numbers that we see. So that's really important. And I mean, if we see that lower respiratory tract infections in itself, that means many viruses are like RSV, influenza, and also together with SARS-CoV-2 during the pandemic, this was one of the leading causes of death worldwide. So we need something there where we believe that we can contribute to find a solution to this. And I mentioned as well, we don't want to go altogether through all the pain of the consequence of the pandemic again, like we experienced for COVID-19. So if we really had something at hand globally, that could be stockpiled as a drug product and be used at the next onset of a pandemic, at least for the initial phase, to keep the wheels turning, so to speak, in the society and to protect the vulnerable patient until we have tailored vaccines available, I think this could have a global impact. And if I think beyond, with our first clinical data, where we showed that our technology gives very good levels of protein production from the mRNA in the nasal cavity. This also opens up to think about new concepts of vaccines, and this would be nasal vaccines, where we could also even think about achieving steroid immunity at the nasal surface and potentially also block transmission, so that the virus does not spread as fast as we observed it during the last pandemic. And this would be also self-administered. You would not need any medical personnel for this, so much easier to use to help. And of course, for the rare disease space, Vera, we not only have primary ciliary dyskinesia and pulmonary alveolar proteinosis, but cystic fibrosis. There is a high unmet need to make an impact. But also alpha-1 antitrypsin deficiency is another rare genetic disease that affects the lung tissue. We know all about idiopathic pulmonary fibrosis, also a rare disease where we need better medication and treatments in the future. Or also, if we think about acute respiratory distress, we don't have really satisfying solutions available right now. So we believe there's a lot of room to make an impact globally over the next decade with what we do.
Great. And I think it's a really good thing that Ethris is focusing both on those like larger, more prevalent lung diseases, but also on rare lung diseases as well. I think that's so important. Just was wondering, with the rapid pace of mRNA innovation happening, how are you managing to keep track of that and follow along with that?
That's a very good question, Vera, because mRNA has such a broad field of applications. And we are always every day tempted to do, let's test this or test this, and that might be an interesting area. So for us, it means focus, Vera. Yeah, so it's really focus on our drug candidates that we are developing now. That's our ETH47 program in asthma, and our program ETH42 for the treatment of primary ciliary dyskinesia. To be very focused on moving these now forward. But of course, we are always open also to work with other partners together to explore further opportunities. I mentioned a few of them. To advance our technology. And then, of course, it is always, we never stop on further improving and advancing our technology platform. So it's another very important focus that what we do at Ethris, so that we can really provide the easiest and best use or convenient use a drug product for our patients in the future.
Great. And just a final question to wrap up. As you look back at your career, is there any advice you would like to give to aspiring researchers and entrepreneurs in the mRNA and just overall in the biotech space?
Yes, the first advice is just get started. If you have an idea, don't think too much of what could go wrong. But just wrap up your sleeves and get started. And I mean, in the end, but that's I think common knowledge is it's not a sprint, but it's like running a marathon, So you really need to think in a context a little bit more long term. And I would even say it's not only a marathon, but it's in the end, it's an Ironman, because you not have only one discipline like running, but as an entrepreneur, there are other disciplines involved as well. And like an Ironman, you have swimming as well, and you have biking as well. So it's quite versatile and broad. But get started. It's a great thing that you can do.
Great. Well, thank you very much, Dr. Rudolph, for sharing all those insights with us today.
Thank you, Vera.
That's the end of this week's episode of the Xtalks Life Science Podcast. If you liked today's show, don't forget to rate, review, and subscribe. Thank you all for joining us, and we'll see you next week. Bye.
To join the Xtalks community, visit Xtalks.com and register for exclusive access to curated content like live webinars, news articles, and educational videos. The views and opinions expressed in this podcast are those of the speakers sharing them and should not be taken as professional advice, nor do they necessarily reflect the policy or position of Honeycomb Worldwide. For further information, email us at podcast@xtalks.com.